Hi again, everyone. Welcome to Chapter 9, Part 1b, the second half of Part 1 in the normal lecture series. We're going to pick up where we left off with respiration. So last time we were in and around this slide right here, and we have three major phases, if you will. The first phase is turning glucose into pyruvate. We're taking a 6-carbon glucose and turning it into two 3-carbon pyruvates. This is called glycolysis and occurs in the cytosol. Glycolysis requires a little bit of ATP and gives up a little bit of ATP, so we have a net. So we invest a little bit of energy and we get some back. And so we're going to get a little bit of ATP out of it. And we're also going to get a few electrons that will end up going on to oxidative phosphorylation. Once the pyruvate is created, it is moved into the mitochondria, and we have to lose a CO2. We oxidize it, and we convert that pyruvate into acetyl-CoA, which is what the entry molecule is into citric acid cycle. Acetyl-CoA. We can actually get acetyl-CoA from glucose. We can also get acetyl-CoA from fats and from protein. So it's another entry into making ATP. Then the third phase is, well, let me get to citric acid first. Citric acid cycle is going to generate most of your CO2. If not, there's a little bit of CO2 here, but most of it's here. I'm going to release a little bit of ATP. We're going to release NADH and FADH2. These are electron carriers. So we're transferring electrons onto NAD and FADH. That electron transfer then makes them into these molecules. It's electron transfer. These aren't being synthesized. They're just being added. Electrons are just being added to them. And then they go and deposit those electrons in phase 3, which is electron transport, which involves pumping, and then chemiosmosis, which is using the gradient from that pump to make ATP. And we should make this the biggest symbol because it's the one that makes the boatload of ATP. But again, you should go check this video out. It's worth your time, at least to help you visualize this process. If you're not visualizing things, it's probably not, you probably don't understand it as well as you probably could or should for the best grade you can earn. It is available on ACE, by the way. In this slide, it may look like I'm jumping right straight to number 3, phase 3. But here's the point that I want to make. Oxidative phosphorylation can generate a huge amount of ATP, but it is not good for rapid replenishment of ATP. It's relatively slow. It demands oxygen in order for it to work properly, and there are times when your muscles are working harder than oxidative phosphorylation can resupply. So we have a different type of phosphorylation of ATP. That's called substrate level, and it's much faster, but it's much less efficient. So we can generate some ATP using certain enzymes that will transfer a phosphate back onto ADP. So we start with ADP, we start with some other precursor, for example, creatine phosphate, and we're going to take that phosphate from the creatine and transfer it onto ADP to make some more ATP. This is called substrate level phosphorylation, and is very, very important because you really only have a couple of seconds of ATP in your cells at any given moment. And so if you do any type of work above and beyond just pure resting, you need a little bit of substrate level phosphorylation to make up the difference for what you can't make quickly enough with oxidative. So substrate level phosphorylation is much less efficient, but with oxidative phosphorylation, you can make a lot of ATP. It just requires a little more time and definitely requires oxygen. So here's the graphic from your book. You have some kind of a substrate with a phosphate on it, and this enzyme is sometimes called a phosphotransferase, but we're not worried about that. We're going to transfer that phosphate off and put it on to ADP, and now we have new ATP that can go out and be used for other things. And I just borrowed this from my physiology class. When muscles are at rest, what they'll actually do is they'll build up those reactants that have phosphate on them. For instance, when we're at rest, we have an enzyme that will take ATP and phosphorylate creatine to make phosphocreatine. And when the muscle starts working, it'll start burning ATP, but then the phosphocreatine will supply additional phosphate so that you can continue to sustain ATP during that process of doing muscular work. So it can go this way, and when we need it, we can go back the other way. This is, if I were to actually show this reaction, it would be the arrow going to the left instead of to the right. And creatine kinase can actually catalyze both. So it allows what we call the need for speed. If we have lots of ATP, we can make phosphocreatine. If we don't have much ATP, we can dephosphorylate creatine to make more ATP. Make sure you get that substrate level phosphorylation down. We're going to actually see that later on when we talk about fermentation. So get to know that term, understand what it means, and see if you can't come up with an example like what I've given you. Let's get back to the process of oxidizing glucose, and we're going to start with glycolysis. So we're oxidizing glucose to make 2 pyruvates. So we're starting off with a 6-carbon glucose, 2 3-carbon pyruvates. Glycolysis means the splitting of sugar. Glyco, sugar, lysis, breakdown or splitting. 2 molecules of pyruvate, so 2 3-carbon pyruvates. This occurs in the cytoplasm and has 2 major times. The first time is when we invest a little bit of energy. We have to overcome what we call activation energy. So we're going to invest a little bit of ATP, and then we're going to get a yield of even a little bit more ATP back. So we're going to invest a couple ATP, and we're going to get a few back, and we'll actually net some excess ATP. Does not require oxygen. Does not require oxygen. So this is part of anaerobic respiration as well. This is one of those steps that involves both aerobic and anaerobic respiration. So here are the basic phases of glycolysis. We have 2 major phases. We have an investment phase where we're basically going to phosphorylate glucose, and that requires a couple of ATP. We phosphorylate glucose. Then when we split glucose, we yield 4. So our net is 2. We invested 2, we make 4, so our net is 2. Okay? Our net ATP production here is 2, and you can see that down at the bottom. We're also going to liberate a few electrons in this process, so we're going to make a couple of NADH, and those are going to move on to the electron transport phase, so they're going to skip the citric acid cycle. And we make a couple of pyruvate, which go into citric acid, and a little bit of metabolic water on the side. But key is that we're going to make a little ATP, we're going to free up a few electrons, and we're going to have some fuel for citric acid cycle. So here's a picture of glucose, and you can see here's the oxygen, and then 1, 2, 3, 4, 5, 6 carbons. 1 prime, 2 prime, 3 prime, 4 prime, 5 prime, and 6 prime carbons. If oxygen is quote unquote the 0, 1, 2, 3, 4, 5, 6. And what I've done is I've circled the 2 areas of carbon, 1, 2, and 3, 4, 5, and 6. That's what's going to yield our pyruvate. So we're going to basically crack it right here at this is a covalent bond right here. Even though it's not a single line, it's showing you a 3-dimensional picture. Imagine this like a frisbee sticking out of the page of the screen, if you will. It's getting bigger as it gets closer to us, and then it gets smaller as it gets further away. In any case, we're going to split right down the middle, and we're going to get 2 pyruvates. Carbon, carbon, carbon there. That way you know what I'm talking about. And there's 3 other 3. So 2 times 3 carbon pyruvates. So after we create pyruvate, we are going to oxidize it further on the way into the inside of the inner mitochondrial membrane. This cutaway of the mitochondrion shows the outer mitochondrial membrane in red and the inner mitochondrial membrane in yellow. So we're gonna send that pyruvate into the matrix, that blue stuff, which is kind of like the cytosol of the mitochondria. In any case, we're going to oxidize it on the way in. It's an energy requiring process, and so we're actually going to create a little CO2, and we're going to clip off one of those carbons, getting pyruvate into the matrix. So here's pyruvate, and here you see the COO. That's called a carboxyl functional group. Sometimes you see a carboxyl says COOH, because oftentimes a proton will be attracted to that negative ion. But in any case, that's a carboxyl group. And what happens is, as we move into the inner mitochondrial membrane, we're going to chop off a CO2. We're going to remove that carboxyl group. In the process, we're going to add a CoA to it. That's a coenzyme A. Okay? And we're going to harvest a pair of electrons off of it. So we're going to clip off the CO2, and that's going to yield some energy. We're going to transfer that CoA onto the remaining acetate. This is an acetate functional group. That gives us acetyl-CoA. In doing so, we harvest a little energy. We get NADH. We got a little bit of CO2 there and a proton. Not too worried about the proton. But that's it. So we're cleaving this off, and that yields the energy that allows us to transfer that CoA onto the acetyl group. So here's the pyruvate. We've got a transport protein that clips off that carboxyl group. We harvest those electrons. We now have NADH that can go shuttle those electrons to electron transport. Coenzyme A, we're going to attach that to the acetyl group, and now we have acetyl-CoA. Citric acid cycle is just a set of small steps designed to allow continual cycling of new carbon into the cycle with carbon dioxide and ATP and electrons out. All right, so one cycle, if we have one pyruvate, that makes acetyl-CoA, that one cycle of a citric acid cycle is only going to yield one ATP. but it's going to yield three pairs of electrons in NADH and another pair of electrons in FADH. We're also going to get some CO2 out of it. This is the primary driver of CO2 creation in our bodies. And don't think for a moment that CO2 isn't as important to life as oxygen. Yeah, oxygen gets all the glory in our bodies, but CO2 helps us keep us in the right pH. Okay? Don't ever forget that. But we're going to make a little ATP. We're going to mostly make electrons for electron carriers. We're not creating the electrons. We're just transferring them onto our carriers. And in the process, we're going to make some CO2 while we're at it. So your job, you need to understand how we get the pyruvate into the matrix of the mitochondrion. But now we're in the matrix of the mitochondrion. We have acetyl-CoA. We don't have pyruvate anymore. We've made CO2. We've transferred that CoA on there. Now we have acetyl-CoA. These two carbons are going to get inserted into the precursor of citric acid. And that's the only step I really, really want you to know, is that very first step. We're going to add those two carbons on there, and we are going to make citric acid. That's the very, very first step. The other thing you need to know about citric acid cycle is that we're going to make CO2. We're going to release some electrons. We're going to make 3 NADHs because we've transferred 3 pairs of electrons onto them. We're going to make 1 FADH2. That's another pair of electrons, and a little bit of ATP in the process. You can get that down, just a little bit of memorization. You should be in fairly good shape. You should be able to recognize what's going on from the symbology. Okay? It's not enough just to have this abstractly incorporated into your head. You need to be able to kind of see what's going on. There are 8 discrete steps, and Nobel Prizes have been awarded for elucidating those steps. Each has its own specific enzyme, and each is a redox reaction. Okay? But the first step is right here. Okay? And these are the terms you need to know. We're going to take acetyl-CoA and we're going to transfer that acetyl group, just those 2 carbons, onto a molecule called oxaloacetate. I love that phrase. It's describing the arrangement of the acetate. Acetyl is only a few carbons long. So oxaloacetate is just describing the structure of that molecule, and it forms citric acid, or otherwise known as citrate. Citrate and citric acid are synonyms, okay? What we're going to do after that is we're going to start making carbon dioxide, pulling electrons off, making, creating electron, reduced NADH. We're going to reduce FADH, make a little ATP, mostly carbon dioxide. So we're going to make 3 NADH, we're going to make 1 FADH2, and each one of these molecules represents a pair of electrons that have been removed from citric acid and then the byproducts of citric acid through that cycle. So here is what you need to know, and you don't have to draw these structures out. You can see oxaloacetate here, and we add that acetyl group. That CoA gets recycled. It helps facilitate the movement of pyruvate into the cell, the matrix. You can see in red, here are those carbons, and you can see they just tack them on right here. We break that double bond gets adjusted, and now we attach those 2 carbons right there. What we're going to do down the line is harvest carbons from these guys, and that will create a new oxaloacetate that we add some new acetyl-CoA to. So that's the first step. That's all you need to know. All the other stuff, not important. Just know the products, be able to recognize the products. Okay? We're making CO2. We're making CO2. We're harvesting electrons. We're harvesting electrons. Harvesting electrons. Substrate-level phosphorylation here. This is substrate-level phosphorylation. Okay? Use up a little water, but no big deal. Okay? But we're harvesting electrons, harvesting electrons, harvesting electrons, harvesting electrons. Think of it in those terms. Ask yourself, what's going on here? That's the first step. I got to know that. Oh, okay, we're harvesting electrons here. Be able to recognize motifs in a chart like this. Make it a lot easier for you. And that's it for part B of part 1, right? Okay. So make sure you get that citric, the key steps. What are we making? How do we get that pyruvate in? Making CO2, harvesting electrons, making 1 molecule of ATP. It's simple stuff. It's just that there's a lot of simple stuff which, with several balls in the air, that makes it tricky. Next time in part 2, we're going to move into what looks like something really scary, lots of redox reactions and things, but we're going to show you electron transport and what the goal of electron transport is, and chemiosmosis over here on the right. The key again is the understanding the simple concepts, what's going on and where. If you can start to recognize patterns, then this will be a lot easier than it might seem. All right, I'll catch you on the next time. Thank you for your time and good luck, and let me know if you have any questions.